So, dear students, today's topic is adjuvants. What are adjuvants? Adjuvant, this particular word has been derived from a Latin word adjuvare, which means to help. So, these are the substances when they are mixed with antigen. If they are mixed with antigen, what they do, and they are mixed and injected with it, they enhance the immunogenicity. Immunogenicity of the antigen. So that is their role. Adjuvants are often used to boost up immune response when antigen has low immunogenicity or when only a small amount of antigens are available. For example, we take one example that for antibody response of mice antibody response, it can be enhanced by immunization with BSA, and this BSA, it can be increased about 5 times when it is mixed with adjuvant. So that is how the immunogenicity of BSA could be enhanced. So what are otherwise there are different effects of antigen. They exert different effects on immune system. One, they enhance the persistence of antigen. 1.1. Then second point is that co-stimulatory signals are enhanced. Co-stimulatory signals are enhanced. Thirdly, they also increase the local inflammation. Local inflammation is increased, and further, non-specific proliferation of lymphocytes is stimulated. So, lymphocyte proliferation is stimulated, which is non-specific. Further, certain examples of adjuvants we take here. One example of such an adjuvant is alum. Alum is aluminium, potassium, sulfate. It prolongs the persistence of antigen. When antigen is mixed with an alum, suppose certain antigen is mixed with alum, it would enhance the persistent injection of this alum. It is mixed with alum. The salt precipitates the antigen. This particular salt, that is alum, it would precipitate the antigen, right? When this is precipitated and this injection of this alum precipitate, it results in slower release of from injection site, so that effective time of exposure that is enhanced, right, to the antigen. It increases many folds from few days, let's say earlier, without any adding of the alum with that of the antigen, the effective time of exposure was, let's say, few days, and then it increases or it is enhanced to several weeks when we mix it with the adjuvant. So this time is exposure time to antigen, it increases, And the alum precipitate also increases the size of antigen. Furthermore, the size of antigen, which was normally, suppose say this was this much, it is increased after it is being joined with the adjuvant, and that increases the chances of it by macrophages for phagocytosis. So this is how it behaves, or how it regulates the antigenicity of the any kind of antigen. Furthermore, there is another example: water in oil adjuvants. They are also used. Water in oil. These kind of adjuvants are also used. Such a type of adjuvant that prolongs the persistence of antigen. And there is a very common type of adjuvant which is used, a preparation which is called as Freyens incomplete adjuvant. This contains antigen in aqueous medium, right? Freyens incomplete adjuvant. It contains antigen, one thing, it would have antigen in aqueous medium. Second, in addition to that, it will have mineral oil and an emulsifying agent. Emulsifying agent. And now how this would behave? These emulsifying agent, for example, it could be minite monooleate, right? Which disperses the oil into small droplets surrounding the antigen. And now antigen is like this in small droplets. This antigen is dispersed like this, and these droplets, these small droplets, is surround the antigen, and then antigen is released very slowly at the site of injection. And this preparation is particularly based on Fluence complete adjuvant, which was deliberately formulated, highly effective adjuvant, which was developed by Jules Freund. It was made by him many years ago, and that contained heat killed mycobacterium. Heat killed mycobacterium as an additional ingredient. And there's a component in the mycobacterial cell wall, which is called as muramyl dipeptide. This component, it is a component of microbacterial cell wall. It activates the macrophages, and making the macrophages more phagocytic, means they become more phagocytic, and that is more effective as compared to that of the incomplete form. Activated macrophages, they are more phagocytic than the unactivated macrophages, and they furthermore they express high level of class 2 MHC. Class 2 MHC molecules, and also the molecules of B7 family. We've already read about this B7 family molecules. And the increased expression of M2 MHC class 2, that increases the ability of antigen presenting cell. Antigen presenting cells, they would be their ability would be increased to present antigen to Th cells. So this is how the adjuvant they act, and this kind. And whereas the other type of molecules which I said, which are B7 molecules, how they behave, B7 molecules. Now Second is the expression of B7 molecules is also increased. B7 molecules on antigen presenting cells, they are present on APC, they bind to CD28, which is a cell surface protein on TH cells. And this stimulates, it enhances the T cell immune response. Thus, antigen presentation and the requisite co-stimulatory signals, usually they are increased in the presence of adjuvant. So, this is the role of adjuvant. And LM and fluence adjuvants, they also stimulate a local chronic inflammatory response that would attract both the phagocytes, right? Local inflammatory response is also increased. Local inflammation is increased, and that would attract one thing, phagocytes, and secondly it would also attract towards the site of injection, the lymphocytes, and this infiltration at the of the cells at the site of adjuvant injection, it results in formation of a dense macrophagic phages rich mass of cells, which is called as granuloma. So granuloma is formed, and because the macrophages in granuloma, they are activated, this mechanism also enhances the activation of T cells further. This granuloma rich in macrophages. This part is rich in macrophages which have been activated. So this is how the role, so different roles are played by adjuvant. There are many other adjuvants which are synthetic also. These stimulate non-specific proliferation of lymphocyte, thus increase the likelihood of antigen-induced clonal selection of lymphocytes. So that is all about the